Hello, and welcome to Industry Innovations, a podcast series from the National Lipid Association that explores groundbreaking advancements in emerging therapies in the field of lipidology and cardiovascular health. I'm your host, Dr. Peter Jones. So today's episode is sponsored by Arrowhead Pharmaceuticals, a pioneering biotechnology company that's advancing a priority RNA interference platform to develop innovative treatments. So it's one that's already delivering some clinical promise, and we'll probably get into some of that today. So I'm joined today by Dr. Bruce Given, who's the Chief Medical Scientist at Arrowhead and a veteran leader in biopharma. Dr. Given's career spans decades in both clinical medicine and the pharmaceutical industry, and I will let him discuss his career a little bit more in detail in a moment. He brings with him not only clinical insight, but also deep expertise in the science and strategy behind this therapeutic development. So today we'll explore the origin and vision of Arrowhead Pharmaceuticals, its small interfering RNA platforms, and where the field of RNA-targeted therapeutics is headed. So Dr. Given, thank you for joining us. So let's start first with your journey, your personal journey. What drew you from academic medicine into the pharmaceutical industry and eventually even to leadership roles in companies like Arrowhead and Incisive?
Okay, well, thank you for the invitation to speak, by the way. So I am Bruce Given. I am a physician, boarded internist and endocrinologist. I started doing research as a freshman in medical school, just sort of happened into it and did bench research all the way through medical school, published. That got me into internal medicine residency at Harvard. There again, I sort of fell into an opportunity to spend most of my last year of residency as really a cardiology fellow, working for Victor Zhao, who has now gone on to be president of the National Academy of Medicine. But Victor, was early in his career as already a noted cardiologist. And I wound up doing, a lot of clinical research on the Clinical Research Center, so human studies, and had a great time doing that. I got recruited back to the University of Chicago to do my fellowship with my mentor, Arthur Rubinstein, who went on to be the chancellor, at Penn, of the medical school, I picked my research back up, not only at the bench, but also, again, started to do clinical research center work, a lot of clinical work. And we published a whole series of papers, New England Journal of Medicine, JCI, places like that. I was chief medical resident and then was on the faculty. And I recognized that the thing I enjoyed doing most was actually clinical research, going bench to bedside, you might say, and doing, really doing things that were at the, tip of the spear and what we were trying to understand in human, medicine and physiology. So I actually surprised myself and left academia and went into industry, first at Schering-Plough and Sandoz, where I learned sort of the basics of doing pharmaceutical medicine. And about 5 years after I left academia, I found myself as the head of U.S. Clinical development for Janssen Pharmaceuticals, which is a big piece of Johnson & Johnson Pharmaceuticals, as you probably all know. And then I did that for 5 years, and then for 3 of those 5 years, I also ran sales and marketing in the U.S. For Janssen Pharmaceuticals. So it was an explosive time of growth. It was obviously, for me, it was sort of drinking through a fire hose to run both R&D and sales and marketing. And I'm a physician. I didn't have sales and marketing training. But then I actually moved over to the diagnostic side and became president of international for J&J's diagnostic group. And that was fascinating. We called it the five continents job. because I had responsibility for basically everything out of North and South America, and we even had one of our diagnostic machines in Antarctica. So we even had business in Antarctica. So it was a 5 continents job. And fascinating to manage people of all those different backgrounds, and to be responsible for business in most of the world. And but I learned two things about myself. I learned first of all that I really missed clinical development of pharmaceuticals. It's such a powerful tool for the advancement of society. And I also figured out I'm a lousy politician. And I was at a level, in big pharma now that everything beyond that was politics. And that was just not my thing. So I left a dream job, and became a public company CEO for a small, company that was developing a drug for pulmonary arterial hypertension at that time. And did that, and then from there got that drug approved, and from there went to be chairman of the board of ICON, which is a large multinational clinical research organization. And was just happy doing all that, and but I was also doing board work, and ran across a guy named Chris Anzalone, who is the CEO of Arrowhead, was then and still is. And I was on a board that was doing the very early work in a very recent Nobel Prize winning technology in small interfering RNAs, or RNA interference is better said. And we got the opportunity to look at buying Roche's assets in siRNA, and they had spent something like $1 billion developing these assets, and Chris didn't have anybody to really do the diligence, so he asked me to do it. I got really fascinated, by what Roche had done. And in the end, when Chris succeeded in buying those assets in late 2011, I came on board to run all of the manufacturing, research and development, regulatory program management, basically everything other than the administrative functions. And from there, we've had just an incredible roller coaster of drug development. We've put probably more than 20 different drugs into the clinic, and I'm helping out at this point as chief medical scientist to hopefully get our first approval, which is in the lipid space. But it's been a great journey, and little did I know that my last job in industry would be my best, and clearly most impactful as well.
Oh, that's an amazing journey. Thank you for sharing that with us. Well, it looks like Arrowhead has sort of had a unique origin story. I mean, how did it really begin, and what now distinguishes it from other biotech platforms?
Well, in its first life, it was founded by a gentleman who fell in love with nanotechnology, and just developed a holding company for all things nanotech. And it was eclectic. They were involved with television screens as well as nanotechnology based biotech drugs. And then when he was ready to turn over, and they reverse merged and were a public company, and the founder was ready to bow out and recruited Chris. And Chris said, well, I love the idea, but I'm only going to do biotech, so we'll get rid of everything else. And he licensed the technology out of Caltech, for delivering, this new idea of small interfering RNAs, which had just won the Nobel Prize a few years earlier. People were trying to figure out how to take this incredible discovery and turn it into human drugs. A little known fact about Arrowhead, actually one that everybody forgets, is that the first demonstration of human knockdown of a gene using siRNA with systemic delivery was actually published, based on the Arrowhead technology in Nature back, I think, in 2009. So we were actually very much at the forefront, although we were a tiny company. But then we bought the Roche assets, and then we bought the Novartis assets, when I had come on board. And since then, together with Alnylam, I would say, our two companies have really opened up the whole field of RNA interference, and to the point now that there are already approved drugs. It's showing its value. It's unique in a lot of ways. And our company is a platform company, which is to say we weren't built around a gene or an idea. We were built around the technology. So we have drugs not only in the lipid field, but the obesity field, liver diseases, pulmonary, muscle, and next year, actually this year, excuse me, this year, we expect to be in the clinic with the very first siRNA drugs delivered into the brain, with from peripheral subcutaneous delivery, which I think will be a massively important thing if we demonstrate that we can truly use siRNA in the brain without having to inject it directly into the lumbar through lumbar puncture. I think we will have made a dramatic advance for the field. So we're a platform company, and we love what we do. We take on the toughest challenges in medicine, and that's sort of our history, or how we're designed. Let's put it that way.
Well, that is really amazing. And you're discussing some of this sort of targeted delivery to specific tissues. I mean, I think some of us in the lipid field are more attuned to hepatic delivery of ASOs or RNAs to the liver, and GalNAc and all that to get it to the liver. But your platform seems to come up with ways to deliver RNA to specific tissues that aren't the liver. So how are you developing this platform to treat this variety of diseases, delivering RNA interference into specific tissues?
Yeah, well, so RNA interference at the beginning, as I said, Nobel Prize winning, discovery, got people really excited. Huge amounts of money came in from big pharma, and then all of a sudden, people figured out they couldn't deliver it. It was a little bit like monoclonal antibodies before they figured out how to humanize them.
Yeah.
Uh, they were so exciting, and then all of a sudden, these mouse monoclonals, created terrible immunologic reactions, and the whole monoclonal antibody field stepped back until they learned how to humanize, and then it became the great technology that it is. Very similar for siRNAs. Antisense was discovered decades before. It is really a first generation technology. Excuse me, single-stranded, substances actually cross membranes very easily, and also get into the nucleus very easily. And so antisense really didn't have to have a delivery technology. siRNAs are double-stranded. It turns out they don't cross membranes well at all. So the only way you can get siRNAs into a cell is to take them in through some transport mechanism. That's why GalNAcs were the first siRNAs, because the liver has this receptor that's high abundance, is a scavenger receptor, and we very quickly figured out how to, well, not very quickly, it took a decade, but after a decade we figured out how to deliver these GalNAc siRNAs to the liver. We, very early on, by 2012, 2013, as we were thinking about things, we were also doing GalNAcs, and we were moving ahead. But we recognized for siRNAs to really, for RNAi to really achieve its full potential, we were going to have to escape hepatocytes and do other things. So we had a skunkworks program going from the earliest days. And we actually learned how to deliver to other tissue types using different ligands and different receptors. And so that's why we're now in muscle, we're now in pulmonary, we're now in adipocytes, we're, cardiomyocytes, and in addition to the liver, and we're about to go into the brain. So this is what's special about Arrowhead, because nobody else has, been able to get into so many different cell types and therefore take on diseases with siRNA. But this was a conscious effort to focus when we were very small, didn't have a lot of money, but we always carved out a piece to do this.
That is fascinating, and that is what a great answer and about that. Now, let's focus a little bit more on where you are with these drugs that are targeting specific disease states. And as I said, most of our audience are cardiovascular specialists and interested in lipids and metabolic diseases. So I know, at least in the lipid field, you've got 3 drugs that seem to be quite far along in their development. I'm pretty sure you're excited about them. So can you maybe give us a little look as to where we are with plozasiran, zodasiran, and olpasiran?
Sure. So the first of those, that we discovered was olpasiran. And most of our audience is probably aware of Lp(a) and is probably aware of olpasiran, that's being developed by Amgen. So that was actually our first subcutaneous delivered siRNA. We recognized that that was going to be a huge marketing effort. We were still a small company, so we licensed it to Amgen. And at this point, they're in their outcome study, to go to approval. They've completed enrollment, so it's now just a matter of collecting, the full set of outcomes that they need, and then, hopefully it will work. And we believe very strongly that Lp(a) will work, and it's going to be a huge advance, I think, in cardiovascular, medical care. And we believe olpasiran's, the best-in-class molecule, and we're so excited about it. But in the meantime, so that was actually our third target, that we had gone after. Our fourth and fifth targets that we went after were actually ApoC3 and ANGPTL3. Both of those are targets that we learned about from genetics, where nature did the experiment of knocking out both of those targets, and patients that were fortunate enough that nature did that experiment for them have incredibly favorable lipid profiles. And those families, for instance, that inherit these are known to have a reduced cardiovascular risk, dramatically reduced cardiovascular risk. So we love it when nature has done the experiment and shows us the way. And by the way, also showed us that those patients didn't seem to pay any price from not having ApoC3 or not having ANGPTL3. So those were our next 2. And the ApoC3 siRNA is called plozasiran. It is for severe, actually all the way from mild to the most severe high triglyceride states. It looks like it works in all of them. The phase 2 data in all of those was positive, and in the most severe space, which is the orphan disease of familial chylomicronemia syndrome, we're actually pending with plozasiran at the FDA, as well as at the European Medicines Agency. But we're also doing work on the much larger group of severe hypertriglyceridemia, sort of the world where fibrates and fish oils tend to get used now. And hopefully at some point we'll also be looking at milder mixed hyperlipidemia, where there may be 20 to 22 million patients in the US, and there's really kind of no good therapies in mixed hyperlipidemia. So that's very exciting for us. Zodasiran, which is aimed at angiopoietin protein like 3, say that really fast, 3 times. Yeah, I don't think that works very well. But ANGPTL3, we're just getting ready to start phase 3. I mean, we're within probably a month or two from treating our first patient in phase 3 in homozygous familial hypercholesterolemia. So obviously the most severe end of the cholesterol, the LDL, spectrum. So we're at both ends right now, and then moving toward the middle with our other programs as we work on them. So very exciting to us. We already have phase 2 data in HoFH that looks very good. So that's an exciting program for us as well. So those are our most advanced. Hopefully plozasiran will wind up getting approved this year. We'll see. We submit, FDA reviews, FDA decides, but we're excited. And we published in New England Journal of Medicine last year on plozasiran twice. Also in JAMA Cardiology and also in Circulation. So there's plenty of research out there for interested physicians, for instance, to look at.
Yeah, this is really fabulous for lipidologists. I mean, most of us do not see familial chylomicronemia syndrome, and so FCS it's rare. It's lipoprotein lipase deficiency, monogenetic, and there is an approved drug already, which is an ASO, olezarsen, but you hopefully will get approved as well. But I think we're most excited about the polygenic severe hypercholesterolemic patient. They still get pancreatitis, their triglycerides are high, but they don't have clinically or genetically defined FCS. We're hoping we can get a treatment for those people because they are such difficult patients to control. So we're definitely looking forward to plozasiran maybe getting down the road there for polygenic severe hypercholesterolemia.
Well, we actually, in our FCS study, we started off, we wrote the protocol to be just genetic FCS. And it was actually the FDA that suggested to us that, there are these patients that essentially phenocopy genetic FCS in the clinic, very high triglycerides, polygenic, not monogenic disease like FCS, but the same hypertriglyceridemia and the same propensity for pancreatitis. And they suggested we include those patients. So our FCS trial actually included both.
Oh, I saw.
We actually consider it, we kind of talk about it as genetic FCS and clinical FCS because they're the same clinical problem in the clinic. And, it's kind of the story, treat the disease, not the genes.
Right.
So it's fascinating because nobody's ever done a study like this before. We did it. We found that both groups responded very well. It'll be interesting to see how this all evolves clinically. But actually, we did study those patients.
Oh, that's fantastic to understand. And the same thing's true again with our audience. Probably the only people who see homozygote FH tend to be the lipidologists, and of course right now we have a monoclonal antibody that's approved, even evenicumab for homozygote FH. It certainly would be really great if we could get a small interfering RNA, much less difficult to utilize, such as
you were talking about a small subcutaneous injection once every 3 months.
Yeah, I mean that would be so much better than the IV monoclonal antibody that we have to use in those patients with homozygote FH. Well, I know that one of the things that I wanted to get into a little bit is that the other part of risks for cardiovascular disease include obesity and steatotic liver disease. And you mentioned that you have a couple of targeted RNA drugs for obesity, and it looks like one is targeted for the adipose tissue specifically, and one is targeted in the liver. Is that correct?
It is correct. And actually, this drug should be, going into humans in a few weeks. The one that's adipose targeted, and it's the first siRNA that's ever been developed, to go into humans, focusing on the adipocyte. We love that, not only obviously do we believe that the target is very, very good, but we have a whole phalanx of other targets in the adipocytes are such an active endocrine organ and so important in fat metabolism. We have a number of exciting targets right behind it. So, and the cool thing about siRNA is once you figure out how to deliver to a certain cell type, you can pretty much knock down any gene that you want to knock down in that cell type. So if it works in this first one, if it delivers well and gives us a knockdown, even if that doesn't turn out to be the best disease target, we have multiple other targets behind it. So that's what makes it so exciting. siRNA, once you get delivered to a cell, only if the target doesn't work does the siRNA not work. It's not like small molecule drugs or even monoclonals. Once you know what you're doing, and again, we focus on things where nature's already done the experiment. Nature already tells us it's good to knock down that target. It makes us much more likely to succeed in creating an active drug than other modalities. So yes, we're targeting actually the same, we're targeting, the same pathway with our first two. The drug that's already in humans and already being tested is targeted to hepatocytes. This is inhibin E, and which codes for a subunit of activin E, and activin E is very important in fat metabolism. It tells adipocytes to store fat. It tells adipocytes, to not break down fat. So it's a big player in obesity, and patients who are lucky enough, people who are lucky enough to inherit non-functioning inhibin E, they walk around like us, look like us, but they have less visceral fat. They're leaner, they have less diabetes. They're just metabolically superior, but otherwise they're just fine. So nature's done the experiment knocking down inhibin E. Now, inhibin E's receptor on the adipocyte is the ALK7 receptor. And that's what we're knocking down in the adipocyte. So what's interesting about that is there are other ligands for that receptor. So when we looked in animal models, in fact, the ALK7 outperformed the inhibin E, probably because there are more ligands. But we're excited about both targets, and basically, let the best drug win. And obesity is so important. And from our perspective, there's so much white space. The GLP-1s, have finally given us something to be excited about with obesity. They're such an important innovation, and we're not trying to replace them. We just see a lot of white space around them. Can we reduce the dose and make them better tolerated? Can we give the weight loss without the muscle wasting? Can we do something that comes after the GLP-1, after people have gone through all to help them maintain their weight loss, as opposed to having to stay on what is not an easy medicine to take for a lot of people. So, we're not trying to replace them. We're just trying to help. And I think obesity is going to be an area of tremendous innovation for the next couple of decades. It's such a hard problem. It's so polygenic. It's got so many social aspects as well. I think this is going to be an area of tremendous innovation for the foreseeable future. Not in really because of the GLP-1s, not in spite of them, and they've opened a door just like the statins opened a door really in LDL, and we believe actually that the ApoC3 inhibitors are opening a door. We've had drugs for triglycerides, but they haven't been very good. We think we're opening the door to really doing something serious about triglycerides as well. So all of this fits with our basic concept of just making the biggest difference that we can. So I think obesity is going to be huge. You mentioned steatotic liver disease. We have two molecules, both of which have shown very interesting results. PNPLA3 siRNA for probably the biggest genetic abnormality associated with steatosis. And then a fascinating drug against a a protein, HST17 beta. Again, nature's done the experiment. When people have poorly functioning HST17 beta, they tend not only not have MASH, but they also tend to not get alcoholic liver disease either. And we developed that drug and licensed that to Glaxo. So, Glaxo is developing, that drug. The PNPLA3 is available for licensing. We don't actually think we have the wherewithal to develop a MASH drug. It's just too big a challenge for us on top of what we're doing in cardiovascular and cardiometabolic. But we have those too. So, again, we look for these, these diseases that nature has led the way, and they're all, they're all in the clinic.
Oh, wow. It's incredible. The pathways that you're doing, the partnerships you're developing, and the collaborations in order to get this achieved. It looks like, we've got a ways to go down the road, 5 to 10 years to get a lot of these things to start to come out to fruition. But I just love where you're headed. Now, knowing that, from a clinician standpoint, excuse me, how do healthcare providers and patients, how do we get them ready to prescribe and accept an injectable for these kind of asymptomatic problems? That's what we're dealing with here. When we talk about risk factors, most of the patients don't know they have these. It's like, oh, well, you got this problem. Here, you need to take this injection for the rest of your life for this thing you don't see or feel. I mean, how do both practitioners and patients get ready for that? Is this even something that is out of the specialist's realm to do with the RNA platform? I just see that as a sort of a long-term challenge. What do you think?
Clearly patients are more used to getting, taking pills. They don't necessarily always do it very well, but they're more used to receiving a prescription and just having pills. I think one thing that's helping us a lot, really ironically, are the GLP-1 agonists, because they have, taught patients that they can do that. And of course, it really helps, if you can put it in an auto injector so the patient doesn't have to see the needle. It helps if you only have to do it once every 3 months rather than once a week or once a day. We all had the experience of how hard it was to get patients to go on insulin, partly not so much because it hurt, but partly because it just, the psychological aspects of seeing that needle and injecting yourself. And that became easier for people with the auto injectors, that they didn't have to really, sort of physically, create a syringe and, fill a syringe and then inject themselves. That's part of it. The nice thing about siRNAs is they tend to be very well tolerated. I don't think we've ever had a patient that I can remember discontinuing any of our trials because the injection gave them problems. So they're small volumes, and they're and again, I think we'll see auto injectors, but it is something that people have had to sort of get used to. And as I said, I think the GLP-1 agonists have gone a long way to ease people's minds that maybe this is something that patients can do and will do. With devices that make it seem, not onerous.
Yeah, I hope so. And I hope that really primary care type of physicians will, get on board with this and talk to patients. And that leads up to, how do clinicians and even scientists, how can they stay up with all these emerging therapies like in the RNA platforms? I mean, how do clinicians and scientists do this? Is it going to be in medical society meetings? Is it going to be reading journals? I mean, where does it come into their
It's going to be
practice?
it's going to be and not or, of course, and the proliferation of meetings, the proliferation of medical journals, all of this makes it so hard. Being in the field of drug development, just to stay up on what's going on, in the areas where I'm so focused is very challenging. And I think AI is going to help us with a lot of this. Actually, I think we're we're going to be able to ask AI to follow things for us that we could never do ourselves. I think it's going to help. But for sure, one of the biggest challenges pharmaceutical companies have, when they're going into white space is how do you educate people, and a lot of physicians now don't want to see sales reps anymore. A lot of them were taught in medical school they shouldn't talk to sales reps, so the sales reps aren't as effective as they were maybe when you and I entered industry. I mean entered medicine,
Right.
and we would talk to sales reps and they'd tell us about what was new, say, hey, we've got a new drug for this, and the information wasn't always perfect, perhaps, but at least we knew something had happened. So, now I think we rely more on things like podcasts, and we rely, more on social media, and rely more on medical education at meetings. We were helped, I suppose, by the requirement, that you need to continue to have medical education to maintain your license. So people do, at least have even a requirement that they go and seek out, new information. But it is a challenge, and it's a challenge that in the industry we meet by just communicating in as many ways as we can.
That is all wonderful, and I certainly hope that what Arrowhead has done and how you have developed the RNA platforms and the diseases that you're looking at are not only just for rare diseases, but are really important across many patients, particularly in the cardiovascular health arena. That I'm just excited about that, and what I hope, are you as excited about the future of precision medicine as I seem to be, and hopefully our listeners are? Where do you see that going?
Yeah, so I just think precision medicine is fantastic because we deal with these complex diseases, and sometimes evolution has already shown us the way. If an individual gene is having a very major impact, either because of its absence or its presence, there's the opportunity to learn from that now with all of the information available in various databases, and our ability to really go into those databases and understand what we're learning. And siRNA, RNAi is a great way to knock down a single either bad actor or even a gene that maybe is operating normally and perhaps was very important for us when we were having food scarcity, but now in a time of plentiful food is actually dysfunctional and harmful, like ApoC3. We'll be able to go after those, and in a very precise way. The problem with small molecules is they never hit just their target; they hit 20 other targets that we don't even know about. Which is why most small molecule drugs come with a lot of adverse effects and don't always work well in the clinic and have a high failure rate when we take them into clinics. Whereas siRNA, I think, will have a much lower failure rate and will be much more precise and tend in the end to have less adverse effects.
That is fantastic. Listen, you have really given us a great discussion, not only about Arrowhead and their pipeline, but also about the RNA interference platform. And I really do appreciate you taking the time to discuss all of this with us. I know the audience really appreciates this. And so, Dr. Gibbon, again, thank you very much for your participation today in this latest podcast of industry innovations from the National Lipid Association. Thank you all.
It was my great pleasure. Thank you.